Okay, this is the meeting on the RNA vaccine world, and we are delighted today to have Professor Joseph Lin from Sonoma State. Dr. Lin got his doctorate from UCSF and is a specialist in immunology and virology, and has agreed to give us an understanding of what this new world of vaccines looks like that we've been talking about so much, what Pfizer and Moderna are working on. So with that in mind, Joe, would you like to share a screen and we'll get started?
Sure. So thank you for having me. It's a pleasure to talk to you guys today. So I'm going to give a little bit of a big picture overview. I know that you have already had speakers on vaccines and probably a little bit about immune responses and whatnot, but I think for those who maybe missed those talks, it might help to kind of tie a few things together. So I'm going to talk a little bit about the brief history of vaccines, just to, again, introduce to some concepts. We'll talk about how current vaccines work. And then towards the end, I'm going to really then focus on the two vaccines that are probably in the news right now the most, what are called RNA vaccines or mRNA vaccines. And also this new vaccine that AstraZeneca has just put out, and this was through the collaboration with Oxford in the UK. And this is what's called an adenovirus vaccine. So we'll talk about what those are and get into basically, I have some of the results from that, the clinical trial, the phase 3 trials. Okay, so in terms of the history, when we think about vaccines, you know, all the credit really initially goes to Edward Jenner. And the story goes that it was his observation that he noticed that dairy maids back in the day, and we're talking about the mid 1700s to late 1700s, but dairy maids were in some way protected from smallpox, right? So this observation led to the idea that, okay, why is there this population that would never get smallpox? And again, during the time, smallpox was a horrible disease. And you also have to keep in mind that nobody really understood what viruses were back then. The concept of a virus was not even around. It was just diseases occurred. But the association here was that, well, dairy maids would sometimes get infected with cowpox. And cowpox was a disease that didn't cause a lot of problems in humans. You would get a little bit of a little bit of a pock mark, but it wouldn't lead to serious complications. For smallpox, about 30% of the people who got smallpox would die from smallpox. And so his kind of association here where people that maybe got cowpox could somehow be protected from smallpox led to this idea that he initially took a boy and purposefully infected him with cowpox, right? He had taken a lesion from a dairy maid and then basically inoculated the little boy. And then a few months later, he purposefully tried to infect the little boy with smallpox. And magically to them, he was immune. And so all of this work was published in the late 1700s. And so this was kind of the first real published example where you could protect an individual from basically the disease of smallpox. Okay, so you know, again, they didn't really know any, obviously, of the molecular mechanisms. The idea of viruses didn't really exist at that point. And so there was a lot of kind of, well, what's really going on here? Especially there was a lot of skepticism from the clergy. This is a historic cartoon where you can see the depicting Jenner inoculating people with cowpox and they would slowly become cows. And so obviously, you know, the belief that this was true took some time, but ultimately, you know, you couldn't argue with the results. It was clear that this strategy worked. It wasn't incredibly well organized in terms of, you know, different regions would get different strains of cowpox to try to protect their populations. And it wasn't really until much later that a lot of the doctors around would try to get organized and try to say, okay, here's the strains we should be using. And so up to that point, it was a kind of a, the Wild West of trying to figure out what could protect people. So um, it's still an important observation here, though, because this led to this idea that we could somehow create vaccines. Again, the term vaccines was originated because this was from the cowpox and Vaca's cow. Vaccinia was the cowpox. But again, the science took a little while to catch up. The observation was clearly there. The results were clearly there, but the reasons for why things were working weren't really there yet. Okay, so to understand a little bit why vaccines work, I kind of just want to introduce the immune system a little bit. And obviously the immune system is an incredibly complex thing, so I don't want to get lost in the weeds too much, but the important parts of the immune system to understand in the context of vaccines is that we break down our immune system into kind of two arms. We have what we call the innate immune system, and we have what's called the adaptive immune system. Now, the adaptive immune system is the part that immunologists focus on in the context of vaccines. Now, in the adaptive immune system, these are just depicting a bunch of the immune cells of the immune system, but kind of highlighted here in the green box, these are the cells that are really important for the adaptive immune response. And the key to the adaptive immune response is that these are the things that have memory, right? So when I talk about memory, this means that your immune system actually can remember all of the different pathogens that it's encountered over your lifetime, right? And so that's an important part of the vaccination process. So if we look at B cells and T cells here, right? So B cells are important because they make antibodies, right? Antibodies are what you hear about in the news all the time. This is what people are looking for, and these are the proteins in our blood that protect us from all the different pathogens out there. And so B cells are important in the immune system. And then the other cells are what are called T cells, and they're broken up into two groups. One is what's called a cytotoxic T cell, and the name is implying that these are cells that kill virally infected cells. So cytotoxic T cells will basically circulate around your body. They will recognize a virally infected cell, and they will directly kill that cell. Now, the other type of T cell is what's called a helper T cell. And a helper T cell basically is around to help organize the immune response. If I go back one slide here real quick, this helper T cell, while it's part of the adaptive immune system, it also helps to coordinate the entire immune system, right? So the B cells and T cells are what drive this adaptive response. And the trick here is that they're a little bit slower in responding, meaning that when you first get infected with a pathogen, your innate system is what first starts. It's very rapid in the order of minutes to hours. Your adaptive system simply takes a little bit longer to get going. We're usually talking about hours to days, right? But the innate system, even though it's fast, the problem there is it does not leave memory behind. It's going to behave the exact same way every single time you see a given pathogen.
Dr. Lin, I'm going to put you on hold. There are some people who are not able to see the slides, and I'm just trying to confirm what's happening here, because a number of people can see them just fine. Okay, I'm going to just type one of the people who says she's had a problem. And
So Joseph, it's a good time for you to take a glass of water.
I'm okay.
okay, I'm looking for someone to tell me what device they're watching on. Is it a phone? And if they are not able to see the slides, okay, are you on a phone or a tablet, or are you on a computer? There's an iPad. Okay, desktop Mac is fine. Okay, I'm going to remove Dr. Lin from the projector. Tell me if that fixes the problem. Okay, KT, somebody tell me if that fixes the problem. Carolyn, KT, Jeff.
Remind them that they're muted, that they have to.
You type a message. Type a message that says fine, or not.
Now, George, I'm seeing you alone with the slides.
That's because I'm speaking. It appears that you need to look at either you're hitting your own share screen or what is called the full setting. Okay. I've had an okay from somebody else. Okay, I'm going to put Dr. Lynn back on. It doesn't appear to have anything to do with this. And okay. Dr. Lynn, why don't you take back on where you are? And I hope most of the people are able to see this.
Okay. Okay. So the adaptive immune system, basically the B cells and the T cells, they're a little bit slower to respond, but the key is that they leave behind a memory. And what that simply means is that if we see that same thing again, then the adaptive immune system can respond much, much more quickly, and it actually responds better, right? And so in the bottom left here, here's kind of just a generic graph here, where you can pretend antigen A is, let's say we see a pathogen, right? It'll take a little while to really develop a good immune response. And then we're going to develop an immune response. Then we're going to clear that pathogen. So this is just showing antibody levels, let's say. And after we clear that pathogen, it goes back down. But the key is it doesn't go back down to where you started. It leaves behind some antibodies. And right, and this is when you hear about these tests that are saying, okay, do you have antibodies for something? If you've never been exposed to something, you won't have antibodies to it. But if you have been exposed to something, then it will leave antibodies behind. And the trick there is that now if you see that same thing again, the antibodies are already pre-existing, so it should protect you from getting sick with whatever. And the thing is, if you see that thing again anyways, your antibodies can actually get even better. So repeated exposure can actually develop a really, really good immune response. And that's why sometimes vaccines require more than one shot, because to get a better response, you might need to see that thing twice. Okay, so the key is the adaptive immune system learns, it has memory, it gets better after each exposure. And so that kind of explains, now we know a little bit about the cowpox, smallpox observation that Jenner made, and that cowpox and smallpox are now both known to be obviously viruses, but they're highly related viruses. And so the idea was that cowpox, which doesn't cause disease in humans, could still develop an immune response against it. And then when you actually saw smallpox, your immune system thought it had seen it previously and had already pre-existing antibodies. Even though cowpox and smallpox aren't exactly the same thing, they were close enough that your immune system didn't really know the difference. And that is basically the fundamental rationale behind vaccine development. If we can introduce something to the immune system that doesn't cause disease, and our immune system mounts an immune response to it, then when we see that thing in real life, we will already have antibodies to it. We will already have T cells that can hunt out those infected cells, and therefore it won't cause us any disease or any real problems. Okay, so what are currently used as vaccines? Well, there are a lot of that. So the history of vaccines, again, goes back quite a ways, but we also know that they usually take a long time to develop. Some of the fastest developed vaccines are usually on the order of 5 years, 6 years. So to get to the point we are right now in less than a year is pretty phenomenal. A lot of the common vaccines that we get now took at least 10 years to develop. And so that's why this is such a big step forward here. And we'll talk about why we can do this so quickly now. But previous to what you guys are hearing in the news now, other vaccines like, for polio or measles and whatnot, they usually fell into a couple different categories. Now, the first type of vaccine is what we call a live attenuated virus. And I put the term live in quotes because we don't really think of viruses as being alive. So live here means replication competent, meaning that if that virus can find a host, it can create more viral particles, right? But if we can attenuate it or weaken it in some way, then even if it infected us, it wouldn't cause disease. So that's the hope. A good vaccine should trick the immune system in able to mount that immune response, but it should not cause disease. So weakened but replication competent viruses are probably some of the best vaccines out there. Other forms are what we call dead or inactivated viruses. These are viruses that are either heat killed or chemically killed and then injected into a person. So they also can mount a good immune response. They're typically not as good as a live attenuated virus, but they're still pretty good. And also the last category there are purified proteins. Sometimes all we need to do is just purify one protein from a pathogen, and that's good enough to develop an immune response. Now, the trouble with at least the live attenuated viruses and even the dead inactivated viruses is you have to grow up a large amount of virus, right? And so, for example, the flu vaccine, all of those flu vaccine dosages are actually grown up in fertilized chicken eggs, right? We infect an egg, then we harvest all the virus, and then we inactivate it, right? And so to do that is incredibly labor intensive, right? Resource intensive, meaning that you have to grow up massive amounts of virus. So when, scientists discover a new virus and you're trying to use some of these old approaches, well, number one, you have to figure out how can you grow large amounts of this virus? Is there any way to figure out how to weaken this virus in any meaningful way? And that's why those sorts of studies, trying to attenuate a live virus can take several years because you have to understand a lot about the subtleties of that virus life cycle. Okay, so, kind of the cartoon diagram here on the left is simply, if we had a virus, we have to know the genes in the virus to understand, well, which part is important for infection, which part is the part that makes maybe people sick. And then if we could figure out which part makes people sick, then try to disrupt that part and try to weaken that virus. Now, again, those are the sorts of studies that take a really long time. But the future of vaccines now, at least what's in the news, are what are called mRNA vaccines or just RNA vaccines, and these adenovirus vaccines. Now, before we can really understand mRNA vaccines, we have to really understand what is RNA itself. So, I think the concept of DNA is pretty familiar to most people. And when you first learn about DNA, the word genome is used kind of synonymously. And so what that really means is that within every single nucleated cell, we have our genome, right? And that is, some people say, kind of the blueprint or the instruction manual for us, right? So since we all originated from a fertilized egg, and that we obviously have, we're multicellular, all those cells originated from that initial fertilization event, which means that in theory, every cell in our body should have the same DNA, right? So the cartoon here, or sorry, the picture here is a mouse, right? If I took a cell out of that mouse, right, and here's a neuron, and I took a different cell out of that mouse that's an immune cell, and I took another cell out of the mouse that's an epithelial cell, in theory, all of these cells, if it came from the same mouse, would have the exact same DNA, right? Since it's the same mouse. But the question is, if they all have the exact same DNA, why do they look different? And why do they do different things, right? Because again, they have identical DNA, but clearly they look different and they're going to do different things. So why is it that that's possible? And that's where RNA really comes into the story. Now, DNA is the master kind of blueprint of everything, but just because the information is there doesn't mean that that information is used by every cell. Okay, so that means that this is kind of how we view, or this is what's called the central dogma, right? We have DNA that can then be used to make RNA, and then RNA can then be used to make protein. And the trick here is that, remember, every cell has the exact same DNA sequence in it, but not every cell will express the genes at equal levels, right? Expression of a gene basically means whether or not that gene is on or off, right? So here's kind of a cartoon diagram where you have gene A, gene B. Gene A and gene B, they have the same amount of DNA in the cell, but they can lead to vastly different amounts of protein. because they can basically be driven by vastly different amounts of RNA. So what that really means is that those cells are different, even though they all have the same DNA, they don't necessarily have to have all of the same genes expressed all at the same time, right? So I kind of write the analogy that, if you go to a store and you buy a piece of furniture that you have to assemble, a lot of times those instructions come in, a lot of different languages. You're only using the ones that you can read. Even though the other information is there, you don't necessarily need it. And so that's the same here. You can use the same analogy for a blueprint, right? A blueprint is equivalent to DNA, but when an electrician looks at a blueprint, he or she might be more focused on wiring, outlets, switches. But if a plumber looks at that same blueprint, he or she might be looking at more pipes and plumbing fixtures and drains, right? So all the information is there in the DNA, but what's actually being used is where that RNA comes into play. Okay, so what does that now mean going forward? Well, let's take a look at a coronavirus itself. Now, coronaviruses itself are fairly simple viruses, meaning that they have this outside, what we call an envelope, right? And within that envelope, there are some proteins. And those proteins are important for actually infecting a cell. So this kind of cartoon diagram, here's the spike protein. That's the protein that you probably hear about in the news the most. So it's actually the protein that the virus uses to enter a cell, right? The cell has a receptor. Everyone has this receptor. It's just that the virus uses this receptor to get into the cell. And once it's in the cell, the virus simply releases its genome, and that the viral genome enters the cell, and then the cell starts making viral proteins, thinking that these are its own cellular proteins. And ultimately, the cell produces all these viral proteins, the virus reassembles, and we simply create new viral particles. Now, the immune system can usually detect the production of viral proteins, and that's one of the ways our immune system sees viruses. It looks for cells that are actually creating viral proteins. So now, what does this have to do with the RNA vaccines? If we look more carefully now, here's a coronavirus again. Here's the actual genome. The genome of the coronavirus itself is RNA. But what the trick here is that instead of taking viral particles and actually trying to inactivate a virus or anything like that, all we really need is one of the genes from the virus. And so in the in the RNA vaccines that you guys are hearing about from Moderna and Pfizer, all they've done is they've taken one gene, and this is the spike protein gene, and they simply take this gene in RNA form and they encapsulate it. They basically put it in what's called a lipid nanoparticle. All that is lipids that encase this piece of RNA. It's there, the lipid part is there to protect the RNA. RNA is very sensitive to degradation. That's kind of why you hear about it having to be stored at these ultra cold temperatures and whatnot. But that lipid kind of shell is also there to help that piece of RNA get inside the cell. Once that RNA gets inside the cell, it's really no different than a virus infecting a cell, right? So the key is, instead of using the entire genome, you only need one gene. So that one gene, if it's introduced into a cell, will cause that one viral protein to be made. And that's enough to mount a good immune response to that one viral protein. There's no chance that this thing will, create a viral particle, because you're not introducing the full viral genome. You're only introducing one gene out of this entire virus. Okay, so the mRNA vaccine in some ways mimics a viral infection, meaning you're delivering this kind of foreign piece of RNA into a cell, but you're only doing it with a part of the virus. There's no need to grow up any virus at all, right? There's no need to study what parts of this virus could make you sick. As long as you have one fragment of that virus, that's good enough to really elicit an immune response here. Okay, so this is actually, then, the big advantage, right? You can simply get sequence from the virus and you can create a vaccine. You don't actually have to have the virus in hand. So when Chinese scientists first sequenced the virus and they posted it online, anyone around the world could just download that sequence and create a vaccine. You don't actually have to have the viral particles in hand. The old-fashioned way to make a vaccine, where you had to grow up the virus, you needed to have that virus in hand to do that, right? So the next question is, how do you actually know if this thing works, right? Obviously, in this day and age, we can't do what Jenner did. We can't try a vaccine out. Challenge trials, which is giving someone a vaccine and then actually seeing if the vaccine worked by purposely trying to infect them, that's not considered ethical here in the US. And so what we do instead is we basically inoculate a huge number of people and then wait to see how many of them get sick, right? So that's the strategy that both Moderna and Pfizer carried out. They did their phase 3 trials. So Moderna had 30,000 enrolled in their clinical trial. Now, the key to any good clinical trial is half the people get the vaccine, half the people have to get a placebo, right? And so obviously the placebo isn't going to protect them. And then they wait. Just in the normal course of everyday living, some of these people will, in theory, encounter coronavirus. And then you basically see, okay, if you get sick, then you test it, confirm it that they really got coronavirus. So they enrolled 30,000 people, half got vaccine, half got placebo. And then after a few months, they saw that they had 196 cases of coronavirus. If the vaccine did not work, you would assume that half of them would come from the vaccine group, half of them would come from the placebo group. But when you looked at the numbers, 185 of the 196 cases actually came from the placebo group, and only 11 actually came from the vaccinated group. So that's really good evidence that clearly the vaccine had some sort of protective effect. And the other thing is then they categorized those that got coronavirus into those that got really sick from coronavirus, or essentially COVID-19. And that of those 30, all of them came from that placebo group. And Pfizer basically had very, very similar results. They had an even larger study at 43,000 people. Half got vaccine again, half got placebo, just like any good clinical trial would. 170 cases, and there 162 came from the placebo group, and only 8 came from the vaccine group. So that sort of data is pretty striking. I think if you would have asked a scientist what's a good target number to reach for a vaccine, someone might have said 70% would be a pretty good number. But when you do the math, both of these trials basically come down to almost 95 or right around 95% effectiveness. So again, quite striking. And the major step forward here was this is simply a piece of RNA that can be created in the lab. There's no need to grow up any virus for any reason. That piece of RNA is made without, cells. It's just made in a lab, and then you package it with some lipids and these lipid nanoparticles, and then you inject it. So that's one of the reasons that these came to market so quickly, because we didn't have to figure out how to grow the virus. We didn't have to grow up large amounts of virus. This can all be done just through knowing the sequence identity. Okay. And I kind of then wanted to transition to the other one that's in the news right now is what's called the AstraZeneca or the Oxford vaccine trial. And they also have just finished their phase 3. They're using a slightly different strategy. And here they're relying on what's called an adenovirus. Now, an adenovirus is a naturally occurring virus that, for the most part, doesn't make people sick. It's a virus out there that sometimes causes like cold-like symptoms. But in general, it's viewed as a harmless virus. Now, what these people have done, the scientists have taken the adenovirus that, again, does not cause any real disease, and they take that spike gene from that SARS-CoV-2 virus, the coronavirus, and they just take that one gene from that virus and they put that gene into the adenovirus genome. And so the benefit of doing this is that by transferring that one gene from the coronavirus into a harmless virus, it doesn't make that harmless virus any worse, but what it does is it gives something for the immune system to see, right? And so here, it's almost analogous to the cowpox, smallpox idea, where we don't use a virus that's really similar to the coronavirus. We create a virus from a harmless virus to kind of look more like coronavirus, and that is enough to develop a good immune response. So when you look at the clinical trials here, it gets a little complicated because I don't know if you guys heard in the news, their clinical trial didn't really go as initially they had planned. So some people got different dosaging. If you look at the actual data, it's roughly around 70% protective. Their trials were a little smaller. But the same sort of thing here. Here, you still do have to grow up large amounts of virus, because here you're growing up large amounts of adenovirus. But adenoviruses are so well characterized kind of in the scientific community that this is not a challenging virus to grow up. And you simply need to know the genome from the coronavirus and just take one of those genes and put it into the adenovirus genome. So again, the results here are promising. There are a few more questions surrounding the AstraZeneca, the Oxford trial, but adenoviruses are also a relatively new platform. There are no currently FDA-approved adenovirus vaccines for humans. There are actually some in the veterinary world, but none in humans. And so, assuming both the adenoviruses and the mRNA vaccines get approved, this will really be the first time these platforms get approved for human use. Adenoviruses have been used in clinical trials before, and that's why they have a little bit of a cloudier past. They were actually used in early HIV vaccine trials that did not prove to work. But because of that, we know a lot about adenoviruses. But again, that also makes it a little bit more complicated because it has sometimes not worked. But based on what the Oxford trial is showing, there clearly looks like something is working here. Okay, so kind of just to summarize the things I've told you about, these are basically two new viral vaccine platforms. The mRNA vaccine, the key to both of these is that they're very quick, right? Once that sequence of a new virus gets posted, you can create a vaccine in the order of days, right? The long part is testing it. You want to make sure it's safe, right? You guys hopefully, I think, have learned about phase 1, phase 2, phase 3 trials a little bit. But once you show they're safe, then you got to show they're effective. And so far, both of these have seemed to be both safe and effective. I think the adenovirus vaccine had a couple adverse reactions, but ultimately it was a very small percentage of people that had adverse reactions. And the benefit of these platforms is they don't require a large production of that original pathogenic virus, right? You don't have to learn how to grow large amounts of coronavirus to generate these vaccines. All you need is that genome sequence. Okay. So the biggest unknown then really is how long will protection last? And that's not known. The current clinical trial participants are going to be monitored for about 2 years. And when they monitor them, they're going to look at the antibody levels to make sure that they're staying high. But that's, again, one of the big unknowns. How long will protection last? That's not necessarily dependent on the platform. There are some vaccines that we currently take that don't last our entire lifetime, right? Some of you guys have probably had to go in for a tetanus booster or something like that, right? And so how long some vaccines last is, again, really just dependent on the specific vaccine. Same thing with how good vaccines are. The polio vaccine is incredibly effective. Other vaccines like the flu vaccine aren't always as effective. So it's just really, you don't know until you try it and then just see how it goes. Okay, so with that, I'm happy to take questions. I think Judy is in charge of questions.
Well, let me, I'm going to grab one off of the chat sequence.
Okay.
Obviously, we're quite interested in what do we know about the application of these various vaccine tests to people of different ages. And of course, our concern is older people and people who have various comorbidities. So what do you know about that?
Yeah, so when they design these clinical trials, they spend a lot of time making sure that the safety and the efficacy is spread across not just different ages, but ethnicities as well, because obviously there's plans to have worldwide dissemination of these vaccines. And so when they do these testings, they keep track of all that data. And so for what I've seen, at least, it shows equal or at least roughly similar protection independent of age groups. They did not test young children in these clinical trials that I know of. And so I think, I can't remember the exact age cutoff, but I know toddlers and infants were not in the trial. But I do know that there were large proportions of people over 65 in these trials. And the fact that, to me, the data is pretty striking that so few people who got the vaccine were came down positive, basically. So a good clinical trial does make sure that their population is spread not just through age, but also, ethnicity and whatnot.
Okay, I want to remind everybody that the way we're doing the questions is type them into the chat, and Judy Coleman, who now will take the first question she's got to pass on to us, will serve as the questioner here.
Okay, here's one from Jeffrey Sheff. Some of the diseases damage is said to be caused by immune storms. How did the vaccine avoid producing these storms?
Yeah, so these were called cytokine storms. These are usually the result of when either the immune system sees way too much of something, but a lot of times when it sees too much of it, it sees it in a systemic way. And what I mean by that is, a lot of times if you have a localized inflammation, it's not a huge deal, right? For example, if you cut your finger and your finger gets a little red and it swells a little bit, it isn't the biggest worry. If that thing then starts to spread much more systemically, then that is a bigger issue. So when cytokine storms occur, it's usually because your immune system is basically working too hard, right? Cytokines are proteins that get produced. That picture that I showed with the immune cells all there, one of the ways they communicate with each other is through these cytokines. So cytokine storms are usually a result of an overactive immune response, and that in a lot of ways makes people sick. So for example, when you get a viral infection and you don't feel well, usually you don't feel well because it's your immune response actually working. And the issue here is that cytokine storms are when the immune system is working too hard. And that's why some of those immunosuppressives like dexamethasone were actually working to help people. Initially, you would think, why would an immunosuppressive help somebody who has a viral infection? It's because these people were doing really poorly because their immune system was working too hard. And so the original question was, why does the vaccine not prevent this? A lot of times it's just because of a dosage issue, right? In a vaccine, we very carefully dose how much you're going to be exposed to. The RNA itself does not replicate. We put a certain amount in, and that's the amount your immune system will see. When you have a viral infection, viruses can replicate. So you might start with a very low amount of virus, but if that virus replicates, you could end up with a huge, what we call viral load, and that can induce a really robust immune response. So the fact that these vaccines are more titrated to a finite dosage would typically mean that you're not going to have that runaway immune system problem here.
Judy?
Okay, from our favorite nurse, Lynn McAleer. A barrier to developing a vaccine for HIV is the frequency that the virus mutates, making a vaccine ineffective over time. What is the frequency of mutation in the COVID vaccine?
Yeah, so even in the short amount of time we have identified, since coronavirus has been identified, specifically the one causing COVID-19, there have been several mutations that have been picked up. And so clearly it is mutating. But the reason the HIV vaccines have failed is only in part due to the mutation rate. There are other factors involved in why it's been really challenging to develop a good vaccine for HIV. But kind of to answer your question is that yes, the coronavirus is changing, but the reality is any virus changes, right? And so I know it's not a very satisfying answer, but it's simply the hope is that the changes that could occur in a coronavirus, you're always at that balance where if that change occurs, it might be beneficial for it to hide from the immune system, but it also might be detrimental for the ability of the virus to continue to propagate itself. So it's always that balance of, yes, it can mutate, but sometimes the mutations that occur actually weaken the virus. And so you're always at that balance. And so, yes, you worry about viruses mutating. In this context, the vaccine is targeted to the spike protein. Now, the spike protein is the protein by which the virus uses to get into a cell. Because of that, the spike protein can't mutate too much, because if that spike protein mutates too much, then it won't be able to get into the cell, right? Remember, the spike protein has to interact with the cellular receptor. And if the spike protein changes so much that it can't react with the cellular receptor, then the virus can't get into the cell. And that's why I'm saying it's always this balance. While yes, it can change to hide from the immune response, it can't change too much that it's to the detriment of its ability to infect and propagate itself.
Judy?
Okay, from Bert Schraga. Some doctors say not to be one of the first.
Yeah, and that's, again, I'm not a medical professional, so I don't give medical advice in that context. It's, so far 70,000 plus people have taken it, and I will say, at least with the mRNA vaccine, there have not really been any unforeseen side effects. Like I said, that the adenovirus, the AstraZeneca vaccine has had a couple that they had to pause their trial for before they could resolve it. But as far as I know, the mRNA vaccines had nothing beyond anything that anyone would normally expect from any vaccine. So whether or not you want to be first in line, again, that's kind of a personal choice. You're balancing your own kind of risk factors there. I have friends that are physicians, and they're all going to be first in line, partly because they're one of the most risk groups, right? They're treating actual COVID patients, so they want, to them, the protection is worth any potential perceived risk that might be out there. So it's a personal choice in terms of what you think your personal risk is going to be.
Judy?
Jeffrey Sheff, is the success rate independent of blood type?
Yeah, so from what I have seen, it seems to be the case. It does not look like blood type. I think early on there were some kind of reports that blood type might play a role in disease severity and things like that. But from what I have seen more recently, that hasn't really, the numbers haven't, as the numbers increase, that didn't seem supported anymore. So from what I have seen, at least, there is not really a correlation with blood type there. There still is a lot of unknowns about, I think, the normal coronavirus infection, meaning that when you look at a panel of, let's say, a few hundred people that have been infected, some people produce really, really great antibodies. Other people don't really produce very much antibodies at all. And that does not correlate to disease severity. Some people have those antibodies last a really long time. Others have, decreasing amounts. So the ability to predict how good of an antibody response, at least from the natural course of infection, that has been a little bit more complicated or still under investigation.
Judy?
And another from Jeffrey. And he's concerned about the low temperature that's required. So if Pfizer needs a minus 80 degrees centigrade, how does it get it from that temperature to your blood and cells without weakening it? And if it weakens, is it dangerous or merely less effective?
Yeah, so the temperatures are storage temperatures. So minus 80 freezers are, obviously, most people people don't have them in their homes, but we have on campus here, we probably have about 8 of them, and a lot of hospitals will have them. That's for longer term storage. Once you thaw that vaccine, it is stable, at least for, I can't remember the actual data on the length of time, but in the course of hours, it's not gonna immediately fall apart. So I think the longer term storage was more of a concern. And there is a difference between the Moderna and the Pfizer there. And that is the thing. So the lipid nanoparticle, that shell that I was talking about that surrounds it, that is what's proprietary here. Most companies won't tell you exactly what's in there. And that is probably why there's a difference between the Pfizer and the Moderna here, because remember, that shell helps protect that RNA. So my guess is that they have slightly different formulations for that lipid nanoparticle shell, and that through their own independent testing, they've seen that one is a little bit more stable than the other. But I think, again, the -80 thing is more of a long-term storage issue. You have to thaw the thing in order to get it into suspension to inject it into a person anyways. And I don't think the vials, I can't remember how many doses per vial they were planning on doing, but it was not a lot. So it would be used up pretty quickly.
May I ask a question? Obviously, we're concerned about our own situation, but the world situation is also quite important. And for many people in the world, they can't afford the high cost that is being talked about for some of these vaccines. Do you anticipate, from what you might know, whether the RNA vaccine or the adenovirus vaccine can come down dramatically in cost over time?
Yeah, in terms of production, RNA is presumably going to be a lot cheaper to make. And so a lot of that time is you know, so a lot of times when you look at the cost of especially drugs, not just vaccines, but more drugs, it's the amount of money used to develop that thing, right? And I think here the development of these vaccines have been relatively quick, and that this platform to develop a vaccine doesn't require the same amount of research that was required in the older platforms for a vaccine. So I think in that, in that way, the cost will be less. But in terms of how countries that are lesser developed with, that don't have the resources to pay for that, that's a harder question. I don't know,
Sure.
are companies going to be subsidized by governments or the WHO or something to that effect? It's hard to know.
Okay. Judy?
And then Philip Herzog is commenting here that by the time the seniors get the vaccine, enough other people will have taken it that we should have a lot of data on what the side effects are.
Yeah, and that's always going to, it's the sort of thing. Right now, like I said, between the two trials, we're at 70,000 plus people, which is a large number. But as more and more people get vaccinated, we'll learn more. And again, who determines what number is enough to get FDA approval? Those are usually done when you apply to do a clinical trial. You have to justify all your numbers. Basically, when these companies said, we want to enroll 40,000 people, they had to justify why that was enough in their eyes. So a lot of these companies have epidemiologists and statisticians that help develop these sorts of studies to make sure that the numbers that they get are going to be statistically significant. And that's why these trials were so big, because they're not challenge trials. You have to wait for normal people to get sick in order to get any data. And so part of this is that we're getting better data because we're having a surge, right? If there was no surge and the numbers were really, really low, then we would actually have less statistical significance to say that the vaccine is actually working.
And that one kind of goes back to Bert's question on don't be the first. Let's see, Evelyn Howard. Is the vaccine 1 or 2 shots do it, or will this be an annual shot?
That's a good question. So the clinical trials were all 2 shots. The issue is, how long does protection last? Now, there are a few things. So I think most people are probably familiar with the flu shot. And the flu shot's a little different here. So the flu shot is different because every year that formulation has the potential to change. So how the flu vaccine works is that basically almost a year before you would get your vaccine, a group of scientists driven by a lot of the different countries around the world, primarily driven by the WHO, they have to predict which strains of the flu will cause disease, right? There are a lot of different strains of the flu circulating around at any given time. They're going to try to predict which ones are going to be the most prevalent one. Because it takes a lot of time for them to produce the actual vaccines, they have to make that prediction, again, almost a year in advance. And sometimes they're right, sometimes they're wrong. But the fact that different strains emerge year after year means that you need to get a flu shot every year, because that formulation changes every year. So when you get a flu shot, it's actually a mixture of 3 different strains, unless you get what's called the quadrivalent. So there's a trivalent and a quadrivalent. Trivalent is a mixture of 3 strains. A quadrivalent is a mixture of 4 strains. And those were the best guesses as to what was going to be the predominant strain that year. Now, again, some years the vaccine is actually identical to the previous year, because the same strains that were dominant the previous year are going to be the same strains that are dominant this year. but that has the potential to change from year to year depending on the variations in the different strains out there. For the coronavirus, although it has picked up a few mutations here or there, the hope is that it's not going to change so much that the vaccine, hopefully the vaccine will still work. But the big question is we don't know how long the protection will last. And that's simply because the vaccine hasn't been around long enough, right? We can predict based on antibody levels and how quickly antibodies decline, but antibodies are only one part of that equation. It's much harder to measure memory T cell populations. It's much easier to measure antibody levels, but it's all just kind of a wait and see how it goes.
Sherry Madgers, how many shots and how far apart?
Yeah, so these were 2 shots that at least the clinical trials were. Apart, I want to say 3 or 4 weeks, but don't quote me on that one. Yeah, I looked at the clinical trial protocol a week ago, but I can't remember exactly for sure. I want to say something like 3 or 4 weeks in between shots.
And from Marilyn Parr, once we finally get the vaccine here in Sonoma County, will we know whether we're getting the Pfizer, the Moderna, or the third?
Yeah, that's a good question. If you go, I don't know what the distribution schedule is going to be like. I don't know what has shipped where. It's obviously a question you could ask the person who's giving you the actual vaccine. They would presumably have the actual vial and it should be labeled with wherever it came from. But I don't know. I don't have that information in terms of what we would get or where it's coming from. But I would bet that the Moderna and the Pfizer one are going to be probably first to hit the market in the US. The Oxford one, I think there were enough questions raised from their clinical trials that I don't know if that's going to see widespread dissemination as quickly as the Pfizer and the Moderna one will.
I might comment, you have to know which one you got because your second one has to be the same one.
Yeah, your second one has to be the same. That is true.
Judy, other questions?
I think we just got some. Let's see. Evelyn Howard saying that she has 2 friends who got the Pfizer shots through Kaiser. And then Julie Linder's question is, what are the typical side effects that people had during the trials?
Yeah, so the ones that were reported were nothing beyond what normal vaccines, soreness at the injection site, maybe feeling kind of, general not feeling well. And so nothing really beyond kind of if you've ever gotten a tetanus shot or a flu shot or something like that, just those sorts of side effects. The side effects that are worrying that showed up, I think, twice in the Oxford trial were neurological in nature. Those are the ones most people worry about because there are some vaccine side effects induce a very temporary kind of neurological phenomena, and that's what people worry about. But as far as I know, that was not really seen with the Pfizer and the Moderna vaccines.
Okay, I'm going to get us toward wrapping up. Judy, any final question you want to have there?
Phil Herzog, why does the second one have to be the same if they are both using the same mRNA?
Yeah, so for consistency's sake, so when the FDA approval in theory comes out, that would probably be part of the stipulation because that's how it was tested. Would it still actually work if you had one shot with Moderna and then the second shot with Pfizer? Presumably it would probably work, but since it was never tested like that, there's no guarantee it would work, and therefore they're not going to say that should be allowed just because it was never tested. But in theory, it should work. But again, why do it a way that wasn't tested? Why do it the way, you might as well do it the way it was tested.
Okay. Professor Lin, I cannot thank you enough for what's been a very fascinating discussion. And hopefully we can come back in 4 months and discuss how wonderful it's been for all of us. Okay. And now we feel free to go out and do things we couldn't do before. And Professor Lynn has said he would hang on for a few more minutes if there were other questions. I'm in fact going to close this out with a piece of advertising I did at the beginning, which is those of you who know the Sunday Symposium. This coming Sunday, we will be having Kayleen Asbo as our lecturer on Beethoven. Kayleen has been in Oakmont in lifelong learning and Sunday Symposium, and everybody thinks she's a great talker. We currently have her lecture on the website. You can see the name of the website at the top, and she will be available for a Q&A Sunday at 11.
And I can tell you, we taped her yesterday. And if you love Beethoven, you're going to want to run out there and listen to him again. And if you've never listened to Beethoven, you're still going to want to do it. She had us, it's an amazing talk.
Okay, great job. I see several thank yous here. And we really appreciate your taking the time.
No problem. Thank you for having me.
Thank you very much. Thank you.
Okay, that's it. We'll end today. And everybody take care of yourselves.